All right, so in this video, we're going to talk about transcription, which is the second part of the central dogma, where we take the DNA message and take part of it, part that for the protein that the cell needs to build, and convert that into an RNA message in the nucleus. So first, let's talk about RNA. RNA and DNA are both nucleic acids, and they're very similar molecules. They both are made of nucleotides. But RNA has a ribose sugar where DNA has a deoxyribose sugar. So basically the difference is one oxygen. They both have nitrogen bases, and most of them are the same. But in RNA, uracil replaces thymine. The other three, adenine, cytosine, and guanine, are the same in both DNA and RNA. So U and A go together, and C and G go together. RNA is a single-stranded molecule, whereas DNA, we know, is a double-stranded molecule. And there are lots of different kinds of RNA, whereas in DNA, it's pretty much DNA. So some examples include messenger RNA, transfer RNA, ribosomal RNA, which are the three main ones involved in producing the protein. And then we have things like small nuclear RNAs, small interfering RNAs, which play important roles in gene expression and gene control, and we'll talk about those at a later time. So let's talk about this process of transcription. So you have two strands of DNA. You have what's called the template strand and the coding strand. So the template strand is the template to make the messenger RNA. It's what the RNA polymerase, the enzyme that builds messenger RNA, uses to read to build the complementary messenger RNA. The coding strand is the strand that's going to have the same DNA code as the messenger RNA, with the exception that T's are going to be replaced by U's in the RNA. Like DNA polymerase, it builds 5' to 3' only. So the transcribed strand is called the template strand. The untranscribed strand is the DNA of the DNA is called this coding strand. And like I said, it has the same sequence as the RNA. So in transcription, we make a complementary RNA strand, but we don't make it for the whole DNA molecule, just for that particular part of the DNA that is needed to make a particular protein or polypeptide. So this happens in something called the transcription bubble. It's going to involve helicase. It's going to involve some of the other things that were involved in replication, but we only unwind part of the molecule at a time. So RNA polymerase, like I said, is the enzyme that makes an RNA molecule using a DNA molecule as a template. So there are three RNA polymerase enzymes. Polymerase 1 transcribes ribosomal RNA genes. It makes the ribosome. The ribosome is a mixture of ribosomal RNA and protein. RNA polymerase 2 transcribes genes into messenger RNA. That's the main one we're talking about here in this process. And polymerase 3 transcribes transfer RNAs, which we'll talk about a little bit later when we get into translation. Each polymerase molecule has a specific promoter sequence that it recognizes and binds to at the beginning of the genes for these particular types of RNA. So in order for the RNA to bind, there has to be a promoter region. That's at the start of the gene. Part of the promoter region is something called a TATA box binding site. It's just an area that has a bunch of A's and T's in a certain sequence. There are transcription factors that also bind that help to increase the rate of transmission or things like that. We'll talk more about those again later. Enhancer regions, again, increase the amount of messenger RNA that's going to be produced, and they're much further upstream from the gene than the promoter is. So they're basically like turning your headlights from low beam to high beam. So what are transcription factors? They form something called the initiation complex. Transcription factors bind to the promoter region, and it's a bunch of proteins that bind to the DNA. We think maybe hormones may act as transcription factors in some cases, and they can turn on or turn off transcription depending on which transcription factors. They also help position and bind the RNA polymerase to the DNA to start transcription. So once the RNA polymerase is bound, it's going to move down the DNA molecule and transcribe the gene by adding the appropriate RNA nucleotides. Reads 5' to 3'. But one of the things that we've learned is that eukaryotes, unlike prokaryotes, in the gene, there are areas that are non-coding. Now, we used to call this junk DNA, but we now know that it does have a purpose. So junk is maybe a poor term to use there, but it's kind of hung around. Eukaryotic genes are not continuous. There are the parts that actually code for the protein, the exons, and then there are the intervening parts, the introns, the junk. So their exons are expressed, introns are in between the exons. That helps you remember and differentiate those two types. So to make a functional protein, RNA polymerase can't just copy all the gene because it's going to have the junk, the in-between stuff, and it's not going to make a functional protein. So the cell has to be able to read and know which ones are the exons, which ones are the introns, and cut the introns out and put the exons together. And that's something called RNA splicing. And that actually allows the DNA molecule to be much more efficient. We can actually code for more than one protein using the same piece of DNA by differential splicing. Maybe taking one group of exons, put them together makes one protein, whereas if you take a different set of exons from that sequence, you can make other proteins. So this is part of what we call post-transcriptional processing. So when RNA polymerase comes in, it transcribes the whole thing, introns and exons. But to make a functional messenger RNA, one that actually makes a functional protein, we've got to do things to the RNA. And one of the things we have to do is cut out the introns. So we call that the primary transcript or the pre-messenger RNA, the thing that's made initially. And then RNA splicing comes in, edits out the introns, makes a mature messenger RNA transcript, which then can leave the nucleus, go out to the ribosomes, and build a protein that works. So the discovery of these introns and exons was done by two different labs, two different people studying two different things. Robert's lab was studying beta thalassemia, which is a blood disease where the beta globin gene has a mutation in it. Sharp's lab at MIT was studying adenoviruses, which cause the common cold, and how their DNA is expressed as well. So it's really important to splice properly, otherwise you're going to lose bits of coding or maybe had some parts that aren't supposed to be coded. So there's really not any room for mistakes. A single added base lost or added in there can throw off the reading frame and affect the entire protein and oftentimes could result in a non-functional protein. So here we've got a gene. Remember, we haven't talked about this yet, but the message is read in groups of 3 called codons. And so each of those 3 codes for a particular amino acid. So if you affect by adding or deleting, you're going to throw off all of the codons from that addition, deletion down the line. So here's what it's supposed to look like in the normal gene. Here we have a mistake in the splicing. One of the G's didn't get cut out. And so now we've changed our reading frame, and you can see we've actually changed the protein significantly. So there are RNA splicing enzymes called small nuclear RNAs and snRNPs, which cut the introns out. So the spliceosome is several of these molecules that come together. They recognize the ends of the introns, and they cut the DNA, or cut the RNA, excuse me, at the border between the intron and exon, and they form a loop and pull the exons together, allow those to combine, and the excides intron loop is cut out. So they call these SNRPs, or the enzymes. So RNA is acting as an enzyme in this case, in the spliceosome. So I mentioned alternative splicing. Produces alternative messenger RNAs from the same gene. You can make different proteins from one gene. When they started the Human Genome Project, there's about 100,000 proteins that humans make, so they assumed there'd be 100,000 genes. But because of alternative splicing, it only takes about 20,000 genes to make 100,000 proteins in the human body. So different segments can be treated as introns and exons. So again, we've always thought finding a gene that's simple. It's the part of a DNA that codes for a protein, but that piece of DNA can code for more than one protein. So defining a gene is getting more and more complicated. Now, cutting the introns out is only part of what has to happen to the messenger RNA before it leaves the nucleus and heads out to the ribosome. One of the things that has to happen is there are enzymes in the cytoplasm that break up messenger RNA. So we need to protect it. So at the tips, there's things added to the messenger RNA to protect them from these enzymes. So there's at the 5' end, there's a GTP cap added, which is a guanine nucleoside, basically, that's added at that end. Poly A tail is added to the other end. Now, the length of the poly A tail is important because the longer the tail, the longer the messenger RNA is going to exist, and the more the protein is going to be produced. So one of the ways we regulate how much protein is made is by the length of the tail. For some genes, you may need to make lots and lots of that protein, so they're going to have a really long poly A tail, maybe 250 adenine molecules. For others, they only need to make a little bit of the protein, maybe only 50 adenines in the poly A tail.